बिस्मिल्लाहिर्रहमानिर्रहीम। अस्सलाम वालेकुम। साइंस एंड नेचर के देखने और सुनने वालों को वेलकम कहती हूँ। अगेन एंड अगेन वेलकम। टुडे द टॉपिक ऑफ वीडियो इज़ कॉलम क्रोमेटोग्राफी। लेट्स स्टार्ट द वीडियो। Preparation of column. A long hot glass tube with the stop collet at the bottom is used as a column depending upon the experimental conditions. Length. The column may vary in their length and diameter. Use a glass tube to about 25 to 30 minutes, 30 centimeter length and 2 to 3 centimeter narrowing down to 5 to 7 mm near the base is used as column. Procedure. Here is procedure. Bird can be used. Tube is used as a column is through washed and dried. It is then fluffed with the glass or cotton wool placed to narrow of the tube, clamped in vertical positions for filling with the iron like alumina and silicate, etc. A slurry is prepared in a suitable solvents such as hexane, petroleum ether and poured into the tube. The solvent is allowed to pass down slowly to let and absorbents settle in the tube. The solvent which flows in clotting in a glass and can be used again to prevent during and cleaning and changing of column. The upper surface shown remains always carried with the solvents right from the beginning of experiment. यहाँ पे हमारी ये वीडियो एंड होता है। कहीं से समझ नहीं है तो आप लोग कमेंट करके पूछ सकते हैं। प्लीज़ 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 आप लोग कमेंट करके पूछा करें, ये मेरे लिए अप्रिसिएशन है। थैंक यू सो मच। अल्लाह हाफिज़।